Hey, Hammy here, coming at you with the last video of Chapter 9 on cell cycle and cell reproduction. This is covering section 9.5, in which we look at prokaryotic cell division or binary fission. In prokaryotic cell division, if you remember what prokaryote means, that means they do not have a nucleus. Okay, so they do have a chromosome that is usually circular. Okay, so they don't have like 46 chromosomes like humans do. They just, the bacteria and some archaebacteria would just have one loop, okay, one loop of chromosomes or one loop of DNA that is the chromosome. It can be about 1,000 times the length of the cell. So they have some protein and enzymes that will take that big loop and fold it all up into a really dense region called the nucleoid region, which means kernel-like. Okay, so if you have your bacterial cell, often near the center of the cell, you get this loop, okay, a loop of DNA all kind of condensed right in the middle. Okay, so it looks kind of like a nucleus, but there's no membrane around it. It's not membrane bound. You just have it all kind of clumped together in a central region. Prior to cell division, it will replicate into, this will split into two rings of DNA so that there's a copy of the chromosome for each of the daughter cells. And these rings will actually attach. One will attach kind of to this opposite cell walls. It'll have a place of attachment there. So that as it splits, as it pulls apart, it will pull the two separate chromosomes apart. This process is called binary fission, of splitting into two, which fission, splitting, binary, two, so that kind of makes sense. And then the two replicated chromosomes, those two loops, are distributed or separated or pulled into opposite ends to the two daughter cells. And because this is one cell splitting into two, and that one cell, okay, equals the organism. Okay, this is actual reproduction. Okay, asexual without sex is just asexual reproduction, because that one mother cell splits into two daughter cells, into two offspring that are genetically identical. Okay, so it's asexual reproduction. It's one bacteria splits and makes two bacteria. E. coli, which lives in our gut, has a generation of about 20 minutes. They under ideal conditions, so enough nutrients and everything. So in 7 hours, you're talking well over 1 million E. coli. Just starting with one E. coli bacteria in 7 hours, you can have over 1 million of them under ideal conditions, assuming that they're dividing every 20 minutes. So that's pretty impressive division rate. Binary fission ensures that the daughter cell is genetically identical to the parent. Okay, so binary fission, we're talking in prokaryotes, some protists and fungi, so algae, yeast, that are single-celled would be going through mitosis, and it would also be asexual reproduction. But they would be eukaryotes that are using mitosis to divide, to make more yeast or to make more algae, to make more organisms. Okay, they're producing two new individuals. In multicellular fungi use mitosis for growth, renewal, and repair. Okay, for making the organism bigger, you add more cells. Or repair, you get a cut or damage. They remove the damaged cells, and you'd use mitosis to replace those cells, to repair that damage. Okay? During binary fission, the chromosome duplicates. It's important to understand that in binary fission and asexual reproduction, they are genetically identical. Okay, the cell will grow, and divide into two, and there's no spindle or anything like that. The chromosome just kind of attaches to the cell membrane, and I'll show you that on the picture here on the next slide. Sorry, first just a little overview of the function Y divide. Okay, in prokaryotes, why divide? Well, because binary fission equals asexual reproduction equals more and more organisms. Okay, in eukaryotes, some of the unicellular eukaryotes, protists and fungi like the yeast, protists like algae, they're using mitosis and cytokinesis for asexual reproduction. In multicellular eukaryotes, they're using mitosis and cytokinesis for development, growth, and repair. Here's the slide I was referring to earlier, just kind of showing the steps of binary fission. So here would be a bacterial cell. You could imagine it would maybe be a Escherichia coli or E. coli. And it's growing. It's an ideal medium, and it's growing. It will almost double the contents of its cell. It will stretch out, okay, and it will also replicate these little chromosomal loops shown here by the red and the purple. Okay, and these things will attach each one to the cell membrane. And as that thing starts to grow, you'll start to get a little furrow. Remember cleavage, furrow. You'll start to get a little furrow forming right here. And it will build new cell membrane and new cell wall across there as it pulls the two chromosomal loops to opposite ends. And once they're to opposite ends and you can get enough cell wall and cell membrane built, boop, okay, it will pop into two identical daughter cells, okay, that are genetically identical to the parent cell. And again, this can happen very rapidly, and that the time that it takes varies depending on the species of prokaryote or single-celled unicellular eukaryote that you're talking about. All right, well, that wraps up Chapter 9. Again, go back, rewatch any videos that you might need to, write down any questions. Feel free to ask me in class. And otherwise, we'll move on to Chapter 10, which is meiosis or meiosis. This is Hammy, out.